Welcome everyone, and thank you so much for attending today's webinar. I'm Kim Merritt, the Director of Office Operations at the American Rose Society. We have a wonderful presentation today by Mr. David Zlesak. After his presentation, we will go into a 30-minute Q&A session. At that time, if you have a question, you will click the little hand icon that is in the control panel on your screen. I will call your name and then you will unmute yourself to ask your question. All right. If you are a consulting Rosarian, attending this webinar today will earn you a credit. So please, if you're watching with someone else, please post in the questions pod who you are watching with so we can make sure we document everyone's attendance today. All right, so at this time, I'm going to hand it over to the President of the American Rose Society, Ms. Diane Summers.
Thank you, Kim, and thank you and welcome to everybody for joining us on this beautiful Saturday morning. It is my distinct pleasure to introduce Dr. David Zlesak to all of you. I've known David for many, many years. I was trying to figure how long, and I don't remember when I met David. It's been that long that we've known each other, and very fortunate that David is in our district of the American Rose Society. Clearly, he's a member of the American Rose Society, and so we get to hear and learn from David often, and I know that you're going to enjoy the program today. David is a professor of horticulture at the University of Wisconsin River Falls, and he teaches courses like on things like plant propagation, woody landscape plants, plant tissue culture. I think he's pretty much known throughout the world for a lot of research that he's been doing on black spot. He also is an avid breeder, and some of you may grow some of his roses, including Above and Beyond, a very popular rose, very, very hardy, as well as it has his Pretty Polly series. I have all of them, and they're just beautiful in my garden. So with that, I would like to introduce to you at this time, Dr. David Zlesak.
Thank you so much, Diane and Kim. It's a pleasure to be with all of you. And the Milwaukee Rose Society was my home club when I was a kid, and I was inspired to breed roses at the age of 13, reading about Will Rattler's rose breeding hobby. So the Rose Society and just wonderful people that love roses have had a big impact on me to continue my love of plants and horticulture, and eventually go to school to study horticulture and then plant breeding and genetics. And over these years, even though at times I've been working with potatoes and lilies for my thesis work, roses have always been the plant that had the top spot in my heart. And so it's just been a joy over these years to be able to be a part of wonderful projects with colleagues to advance our knowledge of disease resistance in roses, other aspects of roses, so we could all benefit and learn together. So today, what I'm excited to share with you about is the advances that we've been making in understanding resistance to black spot disease in roses, some of the tools that are developing that we're able to apply to roses, some genetic tools, and how we could practically do that. So along the way, I'll share a little bit about some technicalities of the tools, but my hope or goal is to make it straightforward. I know sometimes when people hear the name genetics, they begin to tune out, but please don't, please don't. I think with COVID and PCR tests and all that, people have learned to appreciate genetics a little bit more. So as we think about black spot, those of us that live in climates that are humid, we know what havoc it could cause our roses. So when our roses lose their leaves because of this fungal disease, then they just can't make much energy, and we just don't get those beautiful blooms that we long for. Our plant doesn't build and grow in size. So it's a challenge. And over the years, as especially people have a desire not to use as many pesticides, be more concerned about pesticides, want more low maintenance landscapes. The priority by breeders for disease-resistant roses has increased, which is fantastic. And Will Rattler, with his wonderful Knockout Roses, the original, and then the whole series that came out subsequently, have really elevated our expectations for black spot resistance in roses. So when roses can hold on to their leaves, continue to photosynthesize, they'll have that energy to be able to bloom and just be that wonderful contribution that we long for in our gardens. So as we think about black spot disease in general, or any disease in general, what I'd love to do is walk us through the disease triangle. So many of us have heard about this analogy to help us understand disease and how disease builds over time through consulting Rosarian schools and just pathology talks in general. So as we think about disease, we could visualize it as an area of a triangle. And a triangle has three sides. So there's three contributors that if everything is aligned, we're able to get disease. And I love to tease the students in the introduction to plant science class when we have our session on diseases and insects. We start out the day with our normal weekly quiz on the material the previous week. And then I start walking around the room and try to be creepy. And then I ask them, do I put you at ease? And so, some of them, because they know me over the semester, they're like, yes. But then I tease them and say, or dis-ease. So as we think about disease, that means an abnormality in growth and development, either of our human bodies or our plants. And so when plants are experiencing disease, an alteration from normal growth, it could be caused by either a living factor like a fungus or bacteria, or it could be caused by a non-living factor like nutrition that's off, too much or too little of a nutrient, too much, too little of water. So for the disease triangle, we use this analogy for diseases that are caused by a living factor like fungus, bacteria, and the sides of the triangle here, on the upper left, a conducive environment. So for a lot of fungal diseases, bacterial diseases, we need high humidity, free moisture accumulating on the leaves like dew or lots of rain. On the bottom portion of the triangle, a susceptible host. So we need a rose that's able to get that particular disease, such as black spot. So some hosts are more susceptible or less susceptible than others. So some roses more or less susceptible to the disease black spot. And then the other portion of the triangle or side is a virulent pathogen. So what does that mean? A pathogen is a disease-causing organism. So it's that fungus that causes black spot. And virulent means that that form of that particular fungus, of the black spot fungus, is able to cause disease on that host. So virulent, it means that strong, is able to infect and lead to disease. All right, so a conducive environment for black spot, a lot of free moisture. We'll talk a little bit more about that. A lot of diseases in general, the more moisture that we have for root rots, other things, it tends to be conducive for disease to develop. A virulent pathogen, in this case, we'll talk about that fungus in more detail coming up, but we need that fungus that's able to get in and grow within our host, in a susceptible host. So a plant that's able to get that disease. So the longer, let's see. Oh, I'm sorry, my screen just kind of wigged out. Let me see if I can quickly get back to the present mode. Here we go. There we go. So as any side of that triangle, I don't know what, I'm sorry, it's jumpy. As any side of that triangle gets larger. I'm sorry about my screen here. Let's see if I could just keep it here and maybe it'll continue to work. So as the environment gets more conducive, that side of the triangle gets longer and the area of that triangle gets longer and we get more disease. If you have a host that's more susceptible, that side of the triangle gets longer and the triangle gets bigger and we get more disease. So what can we do as rose growers to help control disease? Well, in that upper right-hand triangle, the triangle is shorter or smaller because the lower line is shorter. So we could grow a rose that's less susceptible to black spot. So for instance, knockout or drift rose. In that lower left-hand triangle, we can grow our plants in a less conducive environment, or we could do both. So we might grow our roses in the sunnier area of our yard where there's more air circulation, so the plants could dry off more quickly. So there's things that we can do to alter those sides of the triangle. So having that in perspective can be really, really helpful in terms of managing disease as rosarians. So we can control our environment to some degree. We could choose which roses we grow. One of the things that's really hard for us to manipulate is what black spot fungus blows in, which specific strain or what's called a race of fungus of the black spot fungus blows in. So this is a portion of my rose garden, and this area here is in full sun all day long. And on the north side, there's those arborvitaes. It provides a wonderful windbreak in winter, and then it doesn't shade the garden because it's on the north. So beautiful full sun, great air circulation all day. So as the sun rises in the morning, it warms the plants, it evaporates off that dew more quickly. So, okay, we could grow our plants in greater sun. And when I irrigate, unfortunately I don't have drip irrigation in here, that would be best to keep the leaves dry. I'll irrigate in the morning so it evaporates the free water more quickly. So, okay, we could be aware of our management practices and how it in fact affects the environment, making it hopefully less conducive for disease to develop. We can also change the environment by changing the physical properties, the chemistry that's on the surface of our plants. So we could spray different fungicides that'll help provide a protective layer to our plants from the fungus penetrating, entering, and taking off. So typically we want to do cultural things first, get as far as we can, and then intervene if we have to as we see disease developing. All right, so that pathogen side of the triangle, there's not a whole lot that we can do, I mentioned, but there's a little bit. So as black spot develops on our roses and the leaves begin to fall, the spores are within that material. And if we get a heavy rain, irrigate with sprinklers, that water, the droplets can splash onto that leaf material and get spores up onto the newest growth and continue the cycle of black spot developing and growing, permeating throughout our plant. So what we could do is try to reduce the amount of what's called inoculum, the starting fungal material that'll lead to the propagation of that fungus. So if we rake up, pull off some of those leaves, that'll help slow down the progression of black spot throughout our plant. So that's basically all that we can really do is try to remove some of that pathogen to slow down the developmental rate of that disease progressing. So what holds the most promise, in my opinion, besides getting our cultural management in place, is finding, developing, selecting for our gardens those roses that are the most disease resistant. So here's a picture of a few different roses. The upper right-hand rose is the great classic rose robusta that's defoliating from black spot in the snow spray garden. In the far back is Twin Sisters. It's a hybrid of the fairy and seafoam. And then in the foreground is Wool Rattler's Knockout. So we can see that certain plants have much more natural resistance. They come with a mechanism to fight off that fungus right within their own genetics. So as we begin to understand more about how they do that and how we could strategically breed roses that combine different genes to fortify against black spot, we can make great progress, just like Will has with his knockout roses. We could build upon that knowledge to support sustainable rose growing into the future. All right, so why did I start working with black spots so many years ago? Well, in my Milwaukee area garden, and then the other places where I lived, northern Wisconsin, Twin Cities, black spot is the most problematic disease that I've routinely faced. All diseases, of course, can be problematic, rose rosette, powdery mildew, cercospora, but black spot can lead to quick defoliation of roses. And in black spot, as we mentioned, is so common throughout all the areas of the world where there's a lot of humidity and free water. So some areas of the world, like the Pacific Northwest, down in Texas, you might have a season where it's relatively dry and you don't have very many problems with black spot, but then another time of the year you have your rainy or wet season and it comes up. So it was a problem that I faced, a problem that other people faced. So I really wanted to learn more about what can be done to breed for resistance. And when I was a graduate student at University of Minnesota, Stan Hokinson, the scientist Kathy Zlesak, were interested in doing work with black spot resistance. So even though my project for my thesis was something different, I always loved going and volunteering, helping them, brainstorming together with them. And we've been working on black spot resistance ever since. So one question that I'd love to share here, since my presenter mode or slideshow mode isn't, here we go. We can get back to it. There we go. What part of the plant do we typically find symptoms first? I gave you the sneak preview there. So we typically find black spot developing from the lower portion of the plant, moving up throughout the canopy. And why is that? Well, in the inner canopy of the plant is where humidity-free moisture tends to build. It's shaded in there, it's a little cooler, it takes longer for dew to evaporate. And as we'll talk about in a little bit, black spot needs at least 6 to 8 hours of liquid water for the spores to be able to penetrate and get into leaves. The leaves up on top of the plant are the first to dry off in the morning. There's more wind, and they tend not to be the first leaves then where black spot can get in. As we pull off leaves off of plants growing in the greenhouse and bring them to the laboratory and put them in little humid chambers and put black spot spores on them, actually, it's the youngest leaves that are the most susceptible that get infected first. They're the most tender. It's the easiest for black spot to get in. However, in our gardens, it's the lower portion of the plant where black spot takes hold, and then it moves up through the plant, and it's all because of the environmental conditions, how important that conducive environment is. All right, so what temperature best aids black spot growth? I'll let you think about it. You could write it down on a piece of paper, and then I'll let you know. So is it a cooler temperature, real hot temperature, moderate temperature? Actually, the most conducive temperature is C, room temperature, about 70 degrees Fahrenheit. That's where black spot grows the most prolifically, the quickest, the most freely. So that's perfect for us. So in the laboratory, we're able to do our experiments. We'll pull off leaves of different cultivars growing in the greenhouse where there's no black spot spores floating around. We'll take those clean leaves to the lab. We'll put them in these containers where the humidity is high, add the spores and droplets of water, and we're able to do great research. The cooler the temperature, the slower the fungus grows. And actually, as it gets really, really hot, even if it's a humid climate like South Florida, black spot begins to slow down in terms of its growth just because of the high heat. So in fall and spring in the deep south, where there's good rains and the temperatures are more moderate, that's when you tend to find more black spot. All right, so let's delve a little bit deeper into this fungus, our enemy. Know the enemy. So what is this fungus? How does it spread? What does it look like under the microscope? Well, here are some photographs of the spores. So the name of this fungus is Diplocarpon rosei. And scientists, we're not always very creative. We love to choose names that have meaning, patterns, organization. So rosei, it only infects roses. Okay, so that's the rosei part. Diplocarpon is the genus of this fungus, and there's other members of this genus that infect other crops like sugar beets and such. But this species, Diplocarpon rosei, only infects roses. So that's what obligate to rosa means. Obligate means it only infects roses. These spores have two compartments. So as we look at them, they're elongated with a bit of a line in the middle. So two compartments, diplocarpon. And what happens is that during the growing season, the fungus makes identical genetic clones of itself. So that's asexual, without sex. So it's propagating itself asexually, genetically all the same, all these little spores. And it could go cycle after cycle. The spores will infect leaves. The fungus will grow. It'll produce new sporulating structures and make new spores. I've been able in the lab to go from spore already to new spores being generated within a week. So in real conducive climates, we can go cycle after cycle after cycle, and black spot can really be problematic and spread quickly. So the fungus enters the plant tissue, the rose tissue, when there's 6 to 8 hours of liquid water on the leaves. The spores within that liquid penetrates the leaf, gets in, and the fungus is it's described as being hemibiotropic. So what that means, or what's involved in the process here, is that the fungus penetrates the leaf. It pretends to act somewhat nice to the rose at first, and at about day or two or three, it changes its methodology a bit, and then it just goes all out war against our roses. It begins killing cells. So it gets into our roses, and roses that don't have a really good resistance mechanism or perception of this fungus getting in, if it could sneak in and play nice a little bit, that fungus really gets in, and then things change, and that fungus, this begins killing rose tissue, and we get these dark lesions of dead cells. And then within that material, then eventually the fungus produces the reproductive structures through which more of those spores are generated. When winter comes, fallen leaves and debris on the ground accumulate, filled with overwintering black spot. Come spring, when the rains come again, new growth starts on our roses, rain splashes, the environment's conducive, the cycle begins again. All right, so we've talked about the pathogen. Sounds sad, depressing. Now let's talk about the hope of resistance in our roses. So there are different ways to think about resistance. as plant breeders, as geneticists, pathologists, as the years have gone by, but this basic model still describes it pretty well. There's synonyms that go along with these words, vertical versus horizontal resistance. We'll use these terms, but there's other terms out there like race-specific, race non-specific, quantitative, qualitative, but there's a lot of overlap between those definitions. So what do we mean by vertical resistance and horizontal? And I don't mean the old Twilight Zone TV show. So vertical resistance is what we've especially been working on, colleagues at the University of Minnesota and myself, Stan Hokinson, and different students over the years. And then horizontal resistance is really important too. Will Rattler made great progress with both. And so what does vertical resistance mean? Well, we could think of it with this lock and key analogy. So a rose, if it has a resistance gene, a vertical resistance gene, we could think of it like a locked door. And if a rose has more than one locked door, we could think of them as being in a hallway. For the fungus to get into that rose house, it has to unlock every door before it can get in and cause disease. The fungus has different keys on its keychain. And as we think about this Diplocarpon rosae as a species, this particular kind of fungus, we could subdivide it in what pathologists call races. So each of the different races, and there's 14 different races now, what makes them different are the keys that they have on their keychain. And that means which doors that they can unlock in the rose. Which roses can that race of fungus get into and cause disease on? All right, so in the case of this fungus, it has keys A, B, and C, and this rose has doors A, B, C, and D. The fungus, this form or race of the fungus, can unlock the first 3 doors, but that last door it can't get into. So we don't get any black spot symptoms typically at all. All right, so what can happen is that the fungus periodically can sexually intermate with other forms of the fungus, and we might get a new keychain or a new set of keys reshuffled. And we might have a new form of the fungus that has keys A, B, C, and D. And then suddenly our garden, there was no black spot before on a particular rose, but now, okay, it's going down hard to black spot. And maybe it's because there was some genetic recombination, breeding of the fungus itself in our garden. Typically, more likely what happens is that as we get plants from garden centers from different parts of the country being reshuffled around, they might come with black spot on them. And that black spot then could hop onto our roses in our garden that wasn't in our garden before. And when Vance, a student years ago, looked at different forms of the fungus around the nation, he learned that these different races were really well distributed. And he attributed that to the most likely cause being how freely roses are sold across state lines from nurseries through box stores, independent garden centers far and wide. All right, vertical resistance. So if we can develop roses that have as many locked doors as possible, we can fortify roses then to hopefully not come down a black spot in our gardens. From our tests so far, most of the roses that we've studied have no locked doors at all. Very few of them have a single locked door. And as we begin to characterize those locked doors or those resistance genes, that's where we're able to make a great contribution to the rose community. So we could learn that Bright Eyes, for instance, has door number 4, and another rose over here at George Vancouver has door number 3. and their resistances are a little different. So let's cross those two roses as parents, and then among the babies, hopefully find those that inherit both the door from Bright Eyes and George Vancouver to begin to accumulate these doors. The priority of breeding for disease resistance in roses is relatively recent among the major breeders. Will's been doing it for a few decades, Cordes, others now too, Mayan, but it's relatively recent. So there isn't that many generations or that much understanding yet about these resistance genes. So the time is right to study these roses that seem to have some resistance, characterize them, and then intermate those that we know have different forms of resistance to fortify the new roses in the future. So how do we go about that? Here's a complex table, lots of pluses and minuses, rows and columns. If there's any accountants out there, I bet you're smiling. Love looking at data. So what we have for the different rows here are different roses. And each of these roses, Parisian Charm, Honeybee, Sexy Rexy, Suri, Love and Peace, so on, are a group of roses that are very genetically distinct. And the columns are the different races of black spot. So we have race 1, 2, 3, and then the isolate is the unique form that we actually collected of that black spot that we have in our collection. So for instance, race 3 GVH, it was collected off of George Vancouver in Hastings, Minnesota. So what we do is collect these isolated lesions of black spot from different parts of the country, from different gardens. We make a collection, so we call that an isolate. We isolated black spot off this rose. And then what we do is carefully challenge each of those different roses with that particular isolate. And then minus means that no black spot developed, the rose is resistant. A plus means that, okay, we did get infection, that rose is susceptible. So how we're able to distinguish those different races to know what set of keys that fungus has is that there's a different plus minus pattern in those vertical columns. So if you go to your garden and you collect some black spot and then you challenge all those roses, and if you have the same pattern as race 3, just like we found for that GVH isolate, a plus plus minus 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 plus minus 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 minus, then you know that, okay, I just have another isolate of race 3. I have race 3 in my garden. And a person could actually have more than one race in their garden, and as years go by, different races can come and go, become predominant. So it's always dynamic and changing. So that's how we learn about the different races, how we take a new isolate and put them in a race. If we find an isolate has a pattern that's not on this list, it becomes the next numbered race. So we could keep adding more races. And then these roses, we know, for instance, George Vancouver has that unique pattern of it susceptible to 4 of the races and resistant to all the others. There's some lack door, or maybe more than one, in George Vancouver. And then that gives us a clue that, okay, George Vancouver has some locked door. Let's study it. Let's learn more about it. Surrey has some negatives there too. All right, let's learn more about it. There's other roses like Love and Peace and Queen Elizabeth that's susceptible to everything. Okay, you have no locked doors. All right, so what did Will Rattler do that is so special that led to Knockout? Besides his wonderful passion for roses, love for roses, being the director of Burner Botanical Gardens, knowing that people wanted roses for their yards that were easier to maintain than most of those that he was taking care of at the public garden. He began breeding roses at home, buying plants of those cultivars that were above average for resistance at Burner, intermating them at home. And what he did was bring home black spot from his various trips. So he was bringing home different races of black spot with different keys. And ultimately then in his garden, he had a lot of races. And most all those roses eventually got some black spot because there was at least some race that could get in and unlock those major doors, if there were any doors in his roses. So basically all the roses were getting some black spot. And then he was selecting those roses that got relatively less black spot. That's horizontal resistance. So he opened all the doors, let the black spot burglar in, and then he selected those roses that had less damage from the rose burglar than the other roses. So he was selecting for this quantitative resistance. So it's different degrees of resistance. So a rose falls along a continuum or a range. So he was slowly selecting roses over generation that had less and less black spot development. So when Will's roses go on the market and people grow them in their garden, they might not get any black spot because you're not bringing home black spot on purpose, probably. And so maybe the locked doors are holding. That's great. Vertical resistance. You locked the burglar out. But if you do have a race that gets into your rose, you're going to have hopefully strong horizontal resistance behind that to then hopefully have your rose still hold on to leaves, grow, bloom, and do well. All right, so there's the whole wonderful knockout collection of roses known for greatly elevated black spot resistance. So in the lab, what do we do? Well, in the lab, I take leaves off the plants, take moist paper towel, little sealable clear salad containers we get from the restaurant supply companies, put the leaves in there, and put little droplets that have a known spore concentration of a particular race of black spot. I leave that liquid droplets on for a couple days, and then we dry them off, so well more than the 6 to 8 hours to make sure they get in. And then within 2 weeks, we'll know if we have a susceptible reaction like on the left. There's dead cells there, and then those white things popping out are the reproductive units of the fungus emitting their spores. Or we might have a resistant reaction on the right where we just didn't have that fungus get in. So that's how we go about determining those plus and minuses for those roses that were part of that original collection where we keep going back to identify what race we might have. But then after that, then we could challenge any rose that we're interested in to know, are you susceptible to this race? Are you resistant to that one? And then that helps us get enough information to know, do you have a locked gene? Let's try to learn more about it. Dr. Devner from Germany, Hanover, Germany, he characterized the very first two locked doors in roses. So these RDR genes, so Rosa disease resistance genes. And the scientists were very straightforward. So number one, number two. So number one, he found in a wild rose species, a form of Rosa multiflora. So it's just coming into some of the commercialized cultivars by some German breeders, some Canadian breeders that he shared some of his parental material with. So that's great. We're just beginning to benefit from it. So that's wonderful. So that locked door provides resistance to a subset of those different races. Vance Whitaker was a student of Stan's many years ago. We were grad students together. I loved helping him with his project, learning along the way. That rose in that upper right-hand picture is George Vancouver, and it has that unique plus minus pattern I showed earlier, and so we knew it has at least one locked door. So Vance began to study that for his master's and then PhD degree. So he crossed George Vancouver with a susceptible rose, Morden Blush, which is susceptible to all the races, and he developed a population of 100 or so seedlings. And the goal then is to challenge those seedlings with each of those races that George Vancouver is resistant to and see which seedlings inherited that resistance and which ones didn't. So you have all these siblings. Some have that resistance, that locked door, some don't. And then we could do fancy genetics to figure out what DNA sequence, what region of the genome do all these siblings have that have the resistance that none of the siblings that are susceptible have. So that helps us zero in on what chromosome, where that gene is, and learn more about that vertical resistance gene. So he finished his master's, his PhD in the early to mid 2000s. There wasn't a whole lot of genetic tools available, at least for ornamentals. A lot was being developed for big agronomic crops where there's a lot of money, but it wasn't being translated that well yet to some of these other crops. So he did the best he could with the tools at the time. We knew that there was something segregating that looked like a single gene, just like Mendel with his peas, but we didn't quite know where in the genome it was located, and got as far as we could. In about 2014, this United States Department of Agriculture grant called Crossbreed was funded, and its goal was to support genetic work in members of the family Rosaceae, which includes not only roses, but apples, pears, plums, cherries, strawberries, raspberries, blackberries. So because of our work with black spot and having the tools in place, even though roses aren't the edible fruit like those others there in the marketplace, we were able to be part of this this grant to study disease resistance in roses, because the knowledge could be extended, hopefully, to other rosaceous crops. So by this point, there was a lot more genetic tools in place that we could use in order to figure out, well, what's unique about the DNA of these resistant siblings that the susceptible ones don't have. So Stan and I were part of that, great collaborators, Jim Bradeen at University of Minnesota Pathology Department. So by that point, Vance's original plants and population he generated from crossing those parents were gone. We weren't able to maintain them all with resources over the years. So I recreated the cross, generated a whole lot of new seedlings, and then we began challenging them in the lab, seedling by seedling, race by race, to see which ones inherited the resistance and which ones didn't. So there's resistant and susceptible reactions depicted here. So lots of little boxes, lots of little notes, really good organization, lots of little leaves are washed, put in these little boxes and followed. So what makes the current environment so conducive to doing the genetic work now is a tool that is called a SNP marker. So as you look at the bottom of the screen here, the bottom right, there's a whole bunch of little letters strung out. So plant 1 through 8, and then a sequence, which are all those little letters. A SNP means a single nucleotide polymorphism. So nucleotides are these building blocks of DNA, CGs, A's, and T's. And so they come in that long chain or sequence. And then a single nucleotide polymorphism, so a single spot in the genome depicted there in red, we find a polymorphism or a difference. Poly means more than one. So we see more than one option at that position. So all the roses there share all the same order of those nucleotides in black, but there's that one spot where they could either have, in this case, a C or a T. So what's being held in my hand there, which is relatively small, these days costs about $15,000 to use and work with. There's 96 little blue metallic mirror-like squares And what happens is that you put a DNA sample of an individual plant in each one of those little squares. And what it could do is test for 68,000 of those single nucleotide polymorphisms peppered throughout the whole rose genome across all the chromosomes. I love to think about it like mile markers on a highway. Every mile, there's that little marker with a number on it. So if your car breaks down and you're on the highway there calling AAA or whoever for a tow, if you could give them roughly what mile marker you're at, they could hopefully find you. But there's a lot of space between the mile markers. They'll get to the vicinity and then hopefully get to you. I love to think of these SNPs as mile markers across the whole rose genome. All right, so are there any mile markers that the resistant roses, seedlings, siblings share that none of the susceptible ones have? And that gets us into the vicinity. There's a lot of genes in the area, a lot more work that can be done, but at least gets us in the vicinity to know, is this a different locked door than that other locked door because it's on a different chromosome? All right, there's a difference. So this tool allows us to zoom in portions of the rose genome to be able to document the vicinity of the gene. And then hopefully later we could come up with a real simple PCR test, just like they've done for COVID. When we know that vicinity, we have a little sequence that we could test for, just like they do with the COVID virus. And then we were able to get a cheaper test to know, okay, does this plant have this locked door or not? But this is the pathway to get there. So Jim Bradine in pathology, Stan Hokinson there, Nala and Jason out in Oregon, they were doing the heavy duty genetic work for all of us on the team, the strawberry people, rose people. So we appreciate them. They were doing that heavy duty genomic work, and the rest of us were growing the roses and the other crops and characterizing them for black spot and the fruit characteristics. So we were able to eventually write a paper on it. So this is just a couple pages of that paper to follow up from Vance's work for RDR3, the third black spot resistance gene. In the lower left-hand side there, we know that RDR1 and 2 are both on chromosome 1 of rose. And then, okay, RDR3, it was on chromosome 6. So we know it's a totally different gene in a totally different part of the rose genome. All right, so after that, we knew that Will Rattler's Bright Eyes has a unique black spot resistance pattern. It was resistant to all of the different races except 12. We did the same thing, crossed with Morden Blush. Okay, found a new locked door that became RDR4. So crossed the parents, got a whole bunch of individuals, tested the babies, used that same 68,000 SNP array thing, and we're able to figure out, okay, it's on linkage group 5 or chromosome 5, so it's a totally different gene, a different part of the genome. All right, so that's great. There's now a few different black spot resistance genes, at least 3 and 4, with George Vancouver and Bright Eyes is easily available for breeders. RDR1 from Thomas's work is finally coming out in some cultivars from German breeders. RDR2, Thomas was quiet about, hasn't published on, and I don't know why that is. I think there's some reason that he feels like maybe it's not as unique or legitimate as he thought it was. I'm not sure the details why he's not pursuing it more, but so we at least have 3 out there, so that's great. Since that point with the funding, we're able to chase a unique black spot resistance in lemon fizz that has a unique pattern resistant to everything except race 7. We crossed it with high voltage, characterized all those seedlings again out of lab work. We're struggling a little bit with that SNP array in order to figure out where it is in the genome and if it's a different location and a different gene. We've kept the work going the best we could after that 4-year Ross Breed grant came to an end. Stan had some money. I volunteered a lot of time and resources to keep the plants alive. Thankfully now we have a new funding with that Rose Rosette grant that has a little bit of black spot component to it, which we're so grateful for. It'll continue the black spot work, and serendipitously, Morden Blush has rose rosette resistance. So that's where we can marry these two projects together. So all of the genetic work, those SNP markers that are all generated, 68,000 SNPs being tested for on every seedling of Morden Blush by George Vancouver, Morden Blush by Bright Eyes, and so on. We're able to propagate all those plants, send them to Tennessee, have all that genetic information ready, knowing that Morden Blush has resistant to Rose Rosette, and then maybe we'll find the mile marker for Rose Rosette resistance in Morden Blush. So in this picture, stands on the left, Deanna, she's a new postdoc from France. She did her PhD with Black Spot with Fabrice over there in Angers. And so we're so grateful. She's really good with the bioinformatics like Jason and Nala were. So she's doing that for us now, part of the team. And then there's Karen Hokinson. She's a plant breeder too. Stan and her are married. And then there's Emily, who's a master's student. And then a couple undergraduate students, Kira and her boyfriend, came to help us uncover the roses in the spring. So we keep all these plants alive and going even after we finish the black spot work. This is the Morden Blush Bright Eyes population in the bigger pots in the back there. Many are beautiful. I think some of them should become cultivars. It's gorgeous. All right, so Deanna's funded by this new grant that covers studying rose rosette resistance primarily, and then following up to be able to get those simple DNA tests for breeders, just like a COVID test, to be able to know, does this rose have RDR3, 4? Do these babies combine RDR3 and 4? To do a quick PCR test. So Ramblin' Red also has a unique pattern. And before the last grant ended, we were able to afford and get that SNP work done. And then during COVID, after COVID, we were able to slowly do the pathology work in the lab with those leaves. And Deanna just reported at a meeting in January, we learned it has a unique black spot resistance gene too, and it probably comes from Rosa cordesii, pictured in the lower right-hand corner, which is an ancestor of Ramblin' Red. And so we call it RDR5, and it's on a unique chromosome. Baby Love is something that I did during COVID after we had the funding. Jim Spruill kindly shared that orange seedling of his on the right, which is much more fertile than Baby Love. And it's a seedling of Baby Love, and it has the same unique pattern of Baby Love. And so we're able to use it as a fertile parent with Morden Blush. I generated those seedlings at home and phenotyped them, brought them to school in the lab here at River Falls. And then now at University of Minnesota, Emily, she was my undergrad advisee over here, and Stan's grad student over there now. She was able to do the heavy duty genetics. We were able to recently do that SNP array with it, get enough money to do that. And she's presenting in a couple weeks on this gene, which is a different gene as well. I'm surprised that we're finding different parts of the genome, a different gene every time. It's not just a variation of the same gene. So what do we have right now available to breeders? Well, RDR1 has a unique locked door. Some cultivars coming out of Germany will soon be hopefully available here too that have it. RDR3, we have George Vancouver, and then since we've learned roses like Sunsprite also have it. RDR4, Bright Eyes, okay. Lemon Fizz has something unique that we still have to finish, and it might be RDR7, Ramblin' Red, but 5, and RDR6. So right now, the only gene here that we have a really good test for is RDR1. Thomas Debner has a really good PCR reaction. RDR3, we're in the process right now this fall and winter to to validate and get some primers to be able to do a real effective PCR test. So hopefully soon we'll be able to give breeders tools to test for RDR3. And then all the other genes, we haven't developed a marker yet, a simple DNA test. So that's what this current grant will hopefully fund. So breeders will be able to take advantage and get as many locked doors together as possible. So for instance here, as we look at the different resistance genes across many of the different races here, we can see ones that they confer resistance to and ones that they're susceptible to. So for instance, if we combine RDR3 and 4, we're going to get resistance to all of the different races, at least in our collection. So resistance is dominant, so that's great. So if we combine 3 and 4, as long as one of those has a minus, the baby should be resistant. So we could strategically combine different locked doors to be able to lock out more and more of those races, which is great. So hopefully if we can combine together 3 or 4 different locked doors, we could have a rose that is super durable for resistance, which is great. So to sum things up here, I'd just love to share that this work takes a lot of time and it takes a lot of support. So USDA grants, University of Minnesota, work here at River Falls, wonderful support by Rose Society people, nursery people that shared different black spotted leaves that they mailed to us in the mail for us to characterize and build that collection of different races. Wonderful companies that share different plants with us, so we have more of them. Like Bailey's gives us more and more Morden blush when we need more to culture the fungus on, and Star Roses share a lot of plants, are interested in this work. So we're very grateful. And then now with Deanna and groups all over, groups that develop that 68,000 snip array at Wageningen in the Netherlands, all these people have made this work possible, and hopefully we'll begin to reap the rewards of roses or greater black spot resistance moving forward. All right, so that ends the general presentation, and I look forward to our discussion, questions that you have, suggestions, comments moving forward.
Thank you so much, David. Okay, everyone, it's Q&A time. So if you have a question for David, please look over into the control panel on your screen. There's a little hand icon. Click on it. It will raise your hand, which will alert me to call your name. And then once I call your name, you'll just unmute yourself and ask David your question. Okay, so we have several questions coming up here. Amy Geyer.
Hi, Amy.
Amy, are you there? You'll have to unmute yourself.
Hello, hi.
Hi.
Hello. Hi. So, you know what? I must have did it by accident in the beginning when I was playing around with the screen. But no, I don't have any questions. But it was a really great presentation. Thank you.
Thank you.
Thank you very much, Amy.
Welcome.
Okay. The next person is Carol Wade.
Yes, thank you for the presentation. My question is, I've heard a lecturer say that the more rosebush is resistant to black spot, the more it's susceptible to chili thrips. What's your opinion?
Oh, that's a fantastic question. Thankfully, I don't have chili thrips way up here in the north, but maybe I could share just a little bit of kind of an associated sort of topic. So for us up here in the north, as we have more and more roses at really good black spot resistance, we're seeing more Cercospora. So before the leaves would get eaten by black spot first because it's more aggressive, And then now, because these roses have all their leaves and they're very healthy and they have a lot of energy, the slower growing pathogen can take hold. And now we're able to select for those that have more or less resistance to Cercospora. So I don't know of any, specific relationship between chili thrips tolerance and black spot resistance or not, but I can imagine that if you have a healthier rose that's filled with more photosynthates and sugars because it's able to hold its leaves, that would just make it more delicious and appealing to a chili thrips. As another kind of associated topic here, what Deanna's going to be doing here in the near future, these different roses that we first identified the locked door in, these black spot resistance genes, what she's going to do is inoculate them with the different races. So even if it is a resistant reaction, that's what she's hoping for. She'll put the black spot on there, and then at different time points, she'll take samples of leaves with the inoculum, and she's going to store them. She'll put them in fixative and then stain them so the fungus becomes visible with dyes that make the fungus visible. And she's going to try to figure out how the rose is resisting the fungus, because we suspect that these different genes have different mechanisms by which it limits the fungus. Does a fungus penetrate the cells or not? Is it the cuticle? So I can imagine that there's going to be different means of resistance as well. And we could use that to strategically combine different genes because of the means of resistance. But then that means of resistance could also maybe have interaction with, for instance, an insect. So maybe this pathway towards resistance in black spot might interact with, I don't know, a preference or lack of preference of chili thrips, but this other means of black spot resistance may not. So I'm sorry I don't have a good answer to your question, but a lot of times a good question opens up even more questions. So I love that. Thank you.
Thank you so much. All right, Carol Edwards. Carol, you'll have to unmute yourself.
Okay. Did I unmute myself?
Yes, ma'am.
Okay.
Hi.
Hi. So my question is, can susceptibility to black spot change in a particular region? I'm in the Washington, D.C. area where we are susceptible to lots of black spot. Does it change over time so that your roses could be resistant to a particular form of black spot and look great for a while, but then they become susceptible to some other form of black spot that's moved into the region, and you've got a problem?
Yeah, that's a great question. And yes, so when that happens, when there's variable responses from year to year, that gives us a clue that there's something unique in this rose that may be a locked door. So what does that mean, or why do I say that? So for instance, that George Vancouver rose, what tipped us off on that is that it was grown abundantly in our region, hardy, super healthy, but in certain gardens, it suddenly started going down. And we eventually learned that, okay, eventually in our region, a race that could overcome, that has the right set of keys, that could overcome George Vancouver, started to become more and more prevalent through people's gardens throughout the region, through storms blowing spores and such. So, and then after a rose is susceptible, and that is what led to that rose honeybee. That's part of that one slide. That's part of the collection that we use to characterize what race a rose is in as part of the core group. honeybee, some years it got hit hard by black spot and some years it didn't. And while other roses nearby did. So what I assume is happening there is that a form or a race of black spot, once it comes to our garden, may not be there permanently. Or races, some are more aggressive than others, they compete with each other. So they displace each other over time. So eventually maybe one year you had prevalent in your garden a race that could overcome that rose so it was susceptible. And then next year, a couple years down the road, it's another race of black spot that's infecting some of your other roses. And then a rose that was susceptible now is resistant again. Usually a race, after it comes into our garden or the area, it eventually kind of sticks, becomes somewhat common enough. But maybe in the early steps or stages, as it comes into an area and ebbs and flows a little, we get these experiences like I had with honeybee and maybe you're having with some roses. So another example is that rose, Baby Love. When it first came on the market, people were so excited. It wasn't getting black spot basically anywhere. So breeders used it a ton, which is great. It only has one black spot resistance locked door. And then people started finding in regions in England and then over in the United States, it going down hard. And now we're getting a lot of its descendants because breeders have used it a lot doing the same. So whenever I find a rose that has been really resistant for a long time and then it goes down hard, I want to collect that black spot and I bring it back to the lab and then I test it across the collection and see, well, are you a different race? Because you obviously have a different key in you. Are you a race that we've already characterized in the past? Or maybe you're a new race that came in the area because you're overcoming this rose that so far in our region hasn't been overtaken by a form of black spot yet. And that's how we continually add more and more numbers to the number of races. So it's a long answer and it does happen. Susceptibility, resistance to a particular rose can change from year to year based on what fungus, race of the fungus blows in. Usually the rose is stable in its own genetics. That's not typically changing, but it's what black spot race you have in your garden.
Okay, thank you. So the next question will be for Elena Williams.
Hi, Elena.
Hello, David. How are you?
Good. It's great to be with everybody.
Yes, it is. Say, my friend Sally and I noticed that there was no RDR2 listed in your chart, and we were curious about that.
I am too. And when I asked Dr. Demner, he tries to be really quiet. So I don't know what was going on, but so I try not to probe anymore. So I don't know.
Oh, okay. Well, that's great.
I want to know too.
Thank you so much.
RDR2, at least what I do know and what he published, it's really close on the same chromosome as RDR1. So I'm not sure what the issue is or what, you know, maybe the challenge why he hasn't published on it since, or but we're grateful that there's a number of others.
And we have a few leaves of Baby Love that have a few black spots. Do you want them? You know, I'm out on the West Coast now.
I would love them. That would be really great. And
Okay,
so
we'll connect.
okay, perfect.
Thank you, Elena. All right. And just a reminder for those that came in a little bit late in the webinar, if you are a consultant Rosarian and you are watching the webinar with someone else, please note in the question pod who you are watching with so you can make sure you get credit for attending today. Bruce Monroe.
David, you mentioned that one possibility of resistance was a stronger cuticle, which made it harder for the fungus to penetrate. Would that also help prevent rose rosette disease, that the the mites would have more difficulty penetrating and inserting their the bacterium into the rows?
That's a really great question, Bruce. I'm not sure. I remember a paper in Hort Science, and now Hort Science is open access, so anybody could Google and try to find it without a paywall. But some researchers looked at the wax on knockout, and it has a unique wax deposition, and they think that that may be part of the good black spot resistance. But knockout that does get, you know, rose rosette pretty commonly. So I'm not sure. That's a great question. It'd be great to study that more. It seems like those little mites find a way to crawl in those crevices and somehow take a bite. They're so aggressive, aren't they? And one of the key things for those mites is that spread the rose rosette virus is that they love floral parts, and they love little soft, tender places to crawl into. So the roses that tend to flower a lot and have their sepals and their petals and things like that accessible for them to crawl within and hide tend to be easier to infect. So the roses like knockout that bloom like mad, and that's what we want. We want flowers tend to attract the mites more readily, just because that's where they like to hide in the floral tissue.
The Research Trust is expecting a proposal from you, by the way.
Oh, I appreciate that so much, and I'd be so honored and grateful, yeah, to have some funding to help with getting these markers available to people. It takes a bunch of validation, so we get a simple DNA test, PCR test for each of those RDR genes yet to come. That'd be wonderful. Thank you.
Thank you.
Thank you, Bruce. All right, Christopher Chittumbar.
Yes, hello. I have a question. Enjoyed your talk, David. What do we know about the diplocarbon, the actual fungus itself? Is there anything different about its nutrient requirements that favor its growth? I know the environmental conditions you've talked about, but I was curious about when you grow them, do you actually grow them on the leaves or in culture, or do you have a nutrient support to keep them going?
That's a wonderful question, Christopher. So what we've done over the years is we found that they grow the best on roses. They love roses most of all. When we put them on potato dextrose agar as a nutrient source, they grow really slow, and they just don't grow as well. So we've cultured them continually on roses, and then what we're able to do to store them is take a section of a rose leaf with a lesion, put it in a little tube, and store it under cryopreservation, so liquid nitrogen. And we're able to suspend them in time. And we found that that works better and is easier than growing them on potato dextrose agar or V8 juice and solidified with agar. Thomas Debner, I love Dr. Debner. He is so bright. He's a leader in thinking always like what's next. So Stan and I are plugging and chugging, you know, with more black spot resistance genes, and we're, you know, knowing that has great contributions. Dr. Debner sequenced the black spot fungus genome. He's doing other cool stuff, so he's like way beyond us, and innovation is setting the stage. So he's fully sequenced one of his isolates, and I think there'll be more that we could learn about the fungus itself through that.
Oh, thanks. That's very interesting. I was also curious from a, you know, I mean, you're doing great work on the genetics and inheritance and so forth of different strain varieties. I was just wondering also, has anybody tried to put in external additives? You know, I'm thinking along to see if that retards the growth of black spot on those leaves. I'm drawing analogies. I have a medical background, so I'm thinking about bacteria, where one tests different various antibiotics to see what slows down a particular organism that's pathogenic. In this situation, is anybody trying other things that may be less toxic than what we buy commercially, the sprays and such? Is anybody going down that route, or looking to see if there are other things that might be more effective but less hazardous?
That's a fantastic question, and yes. So in years past, I worked with Jeff Gilman, and what we did was try a number of more natural products. We would spray rose leaves with them preventatively, and then inoculate with black spot. And what we found is that we did have a significant difference, at least statistically, with some of the silicate products, so potassium silicate. And so the silicate accumulates, sometimes you call it liquid glass, but it'll accumulate, strengthening the cell walls. So what we did was a propagation experiment for one of them, where we took leaves that had mild black spot infection of the rose nearly wild, and we had different concentrations of the silicate through our mist system, and as we went up in concentration silicate, the black spot under the mist bench with those roses progressed more slowly, and then the cuttings rooted more abundantly and quicker. And then we also then put it through irrigation water in a nursery setting, different concentrations over a period of time, and then we inoculated the whole plants, and we found a significant difference. But so it's a slight difference, so it wouldn't be as strong of a control as tebuconazole or propiconazole or whatever people like to use.
Yeah. So
go ahead.
No, go ahead.
One other thing that actually helped quite a bit was actually milk. So some of the organic squash growers, we learned about spray a milk solution, and it helped the powdery mildew in their squash. So we took diluted milk. We used, I have to double check, but I think it was 1 part whole milk to 2 parts water, and then I'd spray it to run off once a week, and then inoculated with black spot, and there was some suppression. We followed up trying to think, well, is it the protein lactoferrin that is in milk? But it wasn't. Lactoferrin wasn't different than the water control, so a person could consider a milk spray. So one of the challenges is if you don't get rain and you continually spray milk on your roses, it accumulates, and you get a little mold starting to grow. So so if you do spray milk periodically, after it sits for a while, rinse it off. Some of the proteins stick to the surface, just so you don't get the accumulation. So those are the key things. We tried actually sulfur, which is labeled, so liquid sulfur that you spray on, and at least in our study, it wasn't any different than the water control. We also tried some of the biologicals, I think, was it Bacillus subtilis, and it wasn't much different than the water control either. So the silicates and then also the milk were what showed some promise, at least in our studies.
Thank you very much, Christopher. Okay, so several more questions here. Stuart Dalton.
Yes, I'm in Northern California and actually don't usually have the problem with black spot. I'm a consulting Rosarian, Master Rosarian here. And this year we had a lot because we had a lot of rain. Free water on the leaves as they were leafing out, and for long periods. So it's been very useful, but one of the things I've been researching and using lately is the use of potassium bicarbonate. And based on the last answer and some of the other discussion, I'm wondering about the potassium bicarbonate. It was available as a purchasable thing, and even in nurseries bicarb old-fashioned fungicide, and it is discussed by the University of California as a eradicant for mildew. So I'm wondering if there's been any work done on that. Unfortunately, it came off the market a couple of years ago, I heard, because of supply problems, but you can buy it as a nutritional supplement for people. It's certainly organic, and of course, it's a nutrient with potassium in there.
That's awesome. Yeah. We tried baking soda, sodium bicarbonate in the same experiment and didn't have any difference. And actually, some of the ARS publications, I believe it was from the 1920s, one of the American Rose Annuals, showed that at least sodium bicarbonate didn't affect black spot development. I know it's kind of a general kind of recommendation to use baking soda, but it would be really great to try the potassium, because I haven't read about that or tried the potassium bicarbonate. So I wish I knew more. Thanks for that tip or that lead.
The
The next person is David Bossert.
Hi, David.
Yes. Hello.
David, do you have a question for David? David, are you there?
Oh, darn it.
Yeah, I think we lost him. We'll move on. David Epley.
Hi, how are you today?
Good, good. Thank you so much for being part of the webinar today.
I had a question. Is there any relationship to where these roses are propagated as to their black spot resistance? I've just found I'm in Orlando, Florida, where we're seldomly below 70 degrees ever, and we're always rainy. And I've found if the things I buy, I have to buy either propagated in Florida or Texas, or I've purchased things recently from Oregon, California, and they're just black spot magnets in my garden.
Wow, that's a really good question. What we found for different varieties of roses, so they have their genetics, their characteristic genetics. As we get them and grow them in the greenhouse, there tends to be a little less susceptibility based on the time of the year. And I think it's due to the thickness of the cuticle, a little bit about the physiology. It almost seems like right after winter, when there's a big flush of growth and things are real tender, real actively growing, there's lots of hormones throughout the plant to put energy into making new tissue versus maybe defenses as much. We have a little more susceptibility. It doesn't necessarily make a locked door open or anything, but once fungus gets in, if it does open a door, the rate at which the fungus grows could vary a little bit based on the physiological state of the particular plant, its history, how it develops. So I can imagine roses from other places based on where they're at in their physiology coming to you there in Florida with the heat and everything until it gets in sync with your series of temperatures and seasons, that you might have some remnant physiological impacts on a little greater susceptibility, or in some cases a little greater resistance. So if they're growing, I think maybe in cooler climates, and I could imagine, and they're in the midst of a great flush and putting energy and lots of growth recently, and they come to you where there's a good amount of black spot, that maybe they'd look a little sicker until they get in sync with your season.
All right, thank you, David. Okay, so David Boster, microphone wasn't working, so he posted his question in chat. He wants to know, I am wondering how a variety of black spot is identified. Microscopy, maybe?
That's a great question. So we can characterize black spot differently in terms of genetic sequence, like Dr. Debner's doing. Vance years ago did a little rough DNA marker work across his different isolates of black spot. So the most important thing that we're interested in for black spot, or at least us as rose growers, is what keys does it have on its keychain? What roses can it unlock the doors of? So we're most interested in those keys. So in order to understand which roses that isolate a form of black spot can open, doing that test with that table where there was those number of roses that were the rows, and then the isolate is the column, is the most practical way to characterize the black spot for our purposes. But beyond that, we could do DNA fingerprinting. We could sequence the whole genome of different backgrounds of black spot. They might fall in the same race because they have the same set of keys or genes for causing disease, but they could have different background sequences variation as well. So microscopically, we could look under the microscope and look for those lesions to make sure, is a rose infected or not? Is the fungus able to produce new spores, those little white things protruding out of the old lesion? Look at those spores under the microscope. But the best way to generally characterize a form of black spot, I think, is using that table like we did, and just to figure out, well, what set of keys does this particular isolate have? Because then that'll tell us what roses it can unlock and get into.
Thank you. So, David, we're almost at the 30-minute mark. Do you have a little bit more time to answer a few more questions?
I do, yeah. Yep.
So maybe 10 more minutes, because I know you have to go, but I want to answer some of these questions in the chat. Pam Smith asks, well, she says, thanks, David, for an understandable presentation. Is someone researching Cercospora, I think is how you pronounce it, as we are definitely seeing an increase?
Yeah, that's a great question, Pam, and great to hear from you. It's great to connect with you and Elena and other dear friends I've gotten to know over the years. So I wish that there was more work being done with Cercospora. So part of the challenge is that it's a little more difficult to culture. There was a master's student of David Byrne's, Stella. She's I think a lettuce breeder now in California, but for her master's, she tried to culture the fungus that leads to that leaf spot disease, Cercospora. So part of the challenge to be able to do as much as what we've done with black spot is that it's really hard to preserve and in a controlled way infect roses with Cercospora. So it's hard to characterize different races and then in a controlled way inoculate plants. So I wish more was being done. Dr. Byrne and some of the students, as they have plants in the field, Dr. Wyndham in Tennessee, he has a lot of Cercospora. So as they have roses in the field, they're rating which ones get more or less of that disease, and then try to see what they could learn in terms of the DNA markers, so SNP markers and other DNA techniques, to see if they could find any genes that seem to be associated with resistance. And it seems like even roses that have really strong Cercospora resistance, like Knockout and Goldeneye for me, they get a little bit. So no rose that at least I've grown yet in my garden is totally 100% immune, but there's definitely wide variations between susceptibility. So I'd love for somebody to take the lead in that, find a way to culture that fungus more reliably and push that work forward.
Great. So there's another question by Spencer Dworkin. He says, does different soil mineral compositions affect the individual black spot races' ability to thrive?
That's a great question. I think mineral resources and the overall health of the plant is really important in terms of how it can fight against black spot, how much resiliency it has. So if a plant is already nutrient stressed, and is growing slow and has challenges, I think it tends to be associated with it taking a harder hit from black spot. So some of those roses like Sunrise Sunset that get some black spot, and if it's getting the nutrition it needs, it's growing so aggressively, the new growth is kind of outgrowing it. So it has good horizontal resistance. So if a rose is stressed and just can't, stay a step ahead, I think that makes it even worse. Let me share a quick example of observation. Over the years, when I've crossed the same two parents, one parent has only spring bloom like a wild species, and the other parent is repeat blooming. And amongst the babies, if you get segregation and some of the full siblings are repeat blooming and some of them are spring only blooming, you're going to get a greater amount of black spot. So if the races can get into the rows, get through the locked doors, and you're relying on horizontal resistance, the repeat blooming ones tend to overall get a lot more black spot than the one-time bloomers, because I think, or at least a hypothesis, is that they're using a lot of energy on forming blooms, and they just don't have as many resources to fight the black spot. So I think whatever stresses a rose plant, be it nutrition, be it putting a lot of energy into flowers, and if it is susceptible and the doors are open, I think that'll just exacerbate the black spot. It just makes it easier for the black spot to take off.
Okay. A question from Martha Negley. She says, I have less black spots since using alfalfa foliar spray. Does the health of the leaves make that much of a difference?
That's a great question. And so maybe in the midst of that nutritional connection, so especially as we're relying on horizontal resistance, I suspect it's even more important to have a plant that is really well nourished, that could help. Beyond that, I know back in the 1980s, when I was starting to grow roses in the American Rose Society, American Rose Magazine, there was a lot of advocacy for alfalfa tea sprays, and I did some of those, and I'm not exactly sure what other components may be in some of those teas and things with alfalfa that may also have an effect, but at least nutritionally, I can imagine that helping the rose, and maybe there's more beyond that in terms of some natural chemicals or properties, like I think Christopher questioned before.
Great. So we have time for one last question. I'm going to go back to the phones. John Moe.
John, hello.
John, can you hear us? I'm not sure. His microphone is lit up, but I'm not sure if it's working.
Oh, darn it. Good to hear from you, John.
Okay.
John's a great member of the Rose Hybridizers Association. We've been dear friends for many years.
All right. So sorry, John. We're going to go ahead and move on to Francis Tom.
Hi, Francis.
Francis, you'll have to unmute yourself. Oh, looked like Francis took off his microphone. Okay. All right. James Beardsley.
Hey, Jim.
Hey, David, how you doing?
Good. Thanks for being part of this today.
Of course. I have it's not a disease question. It's a question of what can we as Rosarians do to help you with your research?
Oh, that's a fantastic question. Yeah, I wish I had some really good answers already thought through and ready. I should have been ready for this question. So I'm grateful. In order to continue this work, we're going to need to characterize a lot more roses for their resistance, susceptibility to different races. So in the greenhouse right now, I have about 182-gallon pots here at River Falls, and the goal is to painstakingly challenge them with the different races. So when we come to a point of having the PCR tests that work with the original rose that we found the gene in, and want to validate it on a broader group of roses, that we'd have that infrastructure, that data set in place. So for instance, if a rose that happens to be susceptible also gives us a PCR product that should only be present for those that have the resistance gene, then we know that that PCR test isn't quite good enough. That segment of DNA that we're chasing isn't quite close enough to the gene of interest. So that's what I'm working on right now. And we have a little bit of undergraduate help, but it's a lot. So Bruce before mentioned that the ARS grant opportunity. So I'm going to work on pulling together a narrative and come up with a proposed budget just to have some more undergraduate help to help me wash all those little salad containers and to help with some of the PCR reactions that we'll do in the future. So that's the very practical thing, to plug and chug through this at the moment, to get those really useful DNA markers in place for people to utilize. Beyond that, I reach out to wonderful people that have unique roses, that are ancestors of roses that seem to have these black spot resistance genes, as I try to piece together the history. So if anybody has red Pinocchio and would be willing to share a cutting with me, I'd really be grateful. And if you have all gold, I'd love a cutting so I can get some DNA of that one, because that one seems to be a progenitor of some of the roses. So I might be knocking on some people's doors or try to ask for some of these older roses that are hard to get. And then beyond that, ask for a little funding to help keep these tests going in the greenhouse. Deanna's doing great work. She's funded by that new grant to do the big time genomics bioinformatics work in order to find those SNPs that we could test in the lab here, which is great. So those are my thoughts at the moment. Thank you, Jim.
Is there a way that we as individuals or as societies can contribute to your funds?
Oh, that's very kind. I have a gift account through the university. If you send me an email, which is david.slezak@uwrf.edu, I'll be able to get you that information. That goes into a gift account that then we can use for the work. And because it's a gift account, then the university won't take tax off of it for administrative fees, which is good.
Okay, can you give that email address again?
Sure. My name, David, and then a period, and then my last name, Zlesak, at the acronym here for University of Wisconsin River Falls, which is at uwrf.edu.
Okay, thanks, David.
Thank you, Jim.
Thank you. All right. So at this time, I will turn the webinar back over to Diane Summers. Diane?
Thank you, Kim, and thank you, David, for such a great presentation. And Jim Beardsley, what a great question to end on. How can we support this type of research? As David mentioned, there is the opportunity to fund directly. I know the Greater Milwaukee Rose Society has actually done some of that in the past. And I would encourage others and other rose societies, if they so choose, to do the same. And of course, the Research Endowment Trust, which is one of the American Rose Society Trust supports various types of research for roses across the country. So yes, 2 great opportunities. I want to share with you that the next 4 weeks, we will be busy with the American Rose Society with webinars each Saturday. The next 3 Saturdays, we are doing webinars, a series related to arrangements with roses. This will qualify for credit for our arrangement judges. It will also be a hybrid arrangement school. So those individuals, members of the American Rose Society who are interested in becoming a judge, an arrangement judge, can participate. And then finally, it's a great opportunity for anybody interested in creating beautiful flower arrangements to learn about all of the design concepts to make your arrangements look nicer. So I would encourage you to participate. You can find out about those through the American Rose Society Facebook page, especially if you're not a member. Members, of course, we do send email blasts out related to our programs. And then finally, in August, we will also have a program by Christian Bedard talking about new roses and for the Week's Rose program and what they're doing related to disease resistance as well. So I'm sure those will be great programs over the next few weeks. Again, I want to thank all of our members as well as those who are not members yet of the American Rose Society. I hope you will consider joining our organization. And I want to thank everybody who supports all of the work that we do related to growing roses and to and research to grow better roses. So thank you all. Have a wonderful rest of your weekend, and hopefully we'll see you again next Saturday.